start codon to the gene's stop codon, and a little bit more on either side. And of course, it's complementary to the template strand, the lower blue line you see right there, because it was made by riding along the template strand and making a complementary RNA to it. Now, before the pre-RNA is allowed to leave the nucleus, oh yeah, there's filling in some of the details. First codon very near the 5' end, the stop codon, the poly-A signal very near the 3' end. Before the pre-mRNA is allowed to leave the nucleus, it has to be modified 3 ways. One is called the addition of the 5' cap, addition of what's called a polyadenosine tail, and removal of the introns. And I guess you can make analogies with other things that are manufactured here in our big world, like when they make cars and things. I imagine before the car can leave the factory where it's made, they have to inspect some things and remove some things and things like that. So it's just kind of a processing of the pre-mRNA before it's allowed to leave the nucleus. Let's see, okay, here it's popped off of the gene, but it's still inside the nucleus. Let's look at the 5' cap. There's an enzyme in the nucleus that will go to these pre-mRNAs and add a special guanosine, a special G residue or nucleotide to the 5' end of the pre-mRNA. This blob right there is supposed to be that enzyme, so it just finds pre-mRNAs, clicks onto their 5' end, and adds this special G to it. The pre-mRNA is, of course, made out of RNA, which is G's and A's and U's and C's. And so at first this doesn't seem like that big a deal. I mean, certainly there's lots of G's throughout the pre-mRNA along with A's and U's and C's. And so what's the point adding one more extra G to the 5' end? Well, hold that thought for a minute about what its function is. Let me tell you about its structure. It's not exactly a normal G nucleotide like you might find within the mRNA. It's actually a guanosine triphosphate with, meaning it has 3 phosphate groups attached to it. Whereas usually inside the mRNA, the normal G's that you find as part of an RNA only have 1 phosphate attached to the ribose. So it has more phosphates than a typical G does, is the main distinction. But as for what the function is, hold off on that question for just a minute. The second modification that we get to the pre-mRNA before it can leave the nucleus is this. There's another enzyme that comes along that's going to add some stuff to this end of it, to the 3' end of it. Here comes that other enzyme. And what this enzyme is going to do is it's going to look for that poly-A signal, which remember is AAU, AAA, near the 3' end of the pre-mRNA. And once it finds that poly-A signal, it's going to add a bunch of extra A nucleotides to the 3' end. And that's called making a poly-A tail. All right, here you see that poly-A signal, AAU, A very near the 3' end, but not exactly the 3' end. And then this looks for that signal, docks with the 3' end, and then it starts making, adding a bunch of extra A's onto the RNA. Like that. Now here in my cartoon, I'm just showing 4 A's in the poly-A tail, polyadenosine tail, but that's just so I can fit it onto the screen. In real life, somewhere between 50 and 250 of these A's are typically added onto the 3' end. So, maybe let's pause here for a second. Maybe a way to summarize these last 2 steps is some extra RNA nucleotides that are not present within the gene have been added on to the pre-mRNA. This G right here remember, is not part of the genomic sequence you would find in the actual gene. It was added on after the fact. And likewise, this long stretch of A's right here were added on after the fact. You won't find a long stretch of A's over here in the gene. Here's kind of a review question. Is the poly-A signal in the gene? The AAU, AAA. Yes, it was, because remember the AAU, AAA that signaled for the poly-A tail to be added was in the gene. In other words, you'd find that AAU, AAA right here in the non-template strand. You'd find an AAT, AAA there. That part is in the gene, but the extra A's right there are not, as is this extra G right there is not. Anyway, so what's the point in adding an extra G to that end and some extra A's to the very end? Well, the poly-A tail and the 5' cap do three different things. One of the things they do is allow the mRNA to exit the nucleus and go into the cytoplasm, because the whole point of having a messenger RNA is to bring the codons to the ribosome, so the ribosome has the recipe for the protein. And so that messenger RNA has to get out of the nucleus and go to the cytoplasm. The poly-A tail and the 5' cap are its exit ticket, so to speak, to allow it to exit the nucleus. To make an analogy, some stores you go into, you have to show your receipt before you get to leave the door. That's what these are. They're the receipt for the mRNA before it can leave the nucleus. Another function of the 5' cap and the poly-A tail is this. Oh yeah, let's let it leave the nucleus. There we go. Out here in the cytoplasm, there are some enzymes that digest mRNAs. By digest, I mean degrade them, tear them up into individual RNA nucleotides. In other words, the cell makes some enzymes that destroy messenger RNAs. And at first that seems kind of wacky, because you figure, wait a minute, the cell needs these mRNAs because they're going to be telling the ribosome the instructions for making each of the proteins. Well, that's true, but the cell doesn't want an mRNA sitting around forever. Once the messenger RNA has come out here and a ribosome has read it once or twice or a few times, the cell wants to get rid of it, because just because the cell is needs to make a certain protein at one point in time doesn't mean it always wants to make that protein forever and forever. And so mRNAs are supposed to have a finite lifespan, and for that reason the cell has these enzymes that destroy them, that degrade them within a few minutes of them arriving in the cytoplasm. As I said, they usually live long enough for the ribosome to bind them and make a protein or two off of them. But as it turns out, these enzymes in the cytoplasm that degrade messenger RNAs would act too quickly. They would destroy the mRNAs perhaps before they even found a ribosome. And so to help increase the mRNA lifespan a little bit, that's what the 5' cap and the poly-A tail also do. They protect the RNA a little while from these degrading enzymes. And lastly, the mRNA has to be able to bind onto the ribosome, or maybe I'm saying it backwards. The ribosome has to be able to bind onto the mRNA. And the 5' cap and the poly-A tail help the ribosome to click onto the mRNA. Sound alrighty? Okay, so just to sort of step back for a second, there's the pre-mRNA. It was the thing that was transcribed right off the gene, so it is as long as the gene. If the gene is 8 kilobases, the pre-mRNA is 8 kilobases. If the gene was 20 kilobases, the pre-mRNA is 20 kilobases. But before the pre-mRNA can leave the nucleus, it's got to have that 5' cap added and have that poly-A tail added. There's a third thing that also has to happen to it before it can leave the nucleus, and this is called removal of introns. And this is, I think, the most fascinating of these three processing steps that have to be done to it. So let's look at what those introns are all about. And the concept of introns can be a little bit confusing. And just to make it maybe a little bit clearer, let's just say for the sake of argument that we're going to look a little bit more closely at this messenger RNA. And this is going to be the protein that it's supposed to encode. Because remember, the whole point of making mRNAs is that they contain codons that go out to the ribosome, and so the ribosome will be able to use the genetic code to make the correct protein. All right, so there's the protein that's supposed to be made from this messenger RNA. I guess it's 7 amino acids long, and there's the actual messenger RNA itself. Now, we've talked about the genetic code. Here it is, where each amino acid is represented by these codons, these 3 consecutive letters in the mRNA, and this is how the ribosome decodes the mRNA. Now, the first amino acid of this particular protein is methionine, MET for short, and the codon for that is AUG. And so we should be able to find the first codon in the messenger RNA if we go looking for it. And I guess there it is right there, AUG. And so as far as the ribosome is concerned, that makes total sense. There it is, first codon. And so that's how the ribosome knows that the first amino acid is supposed to be the methionine. Now, the second amino acid in this protein is arginine, and the ribosome is going to know that because we're expecting the codon number 2 to be a codon for arginine. And here we see the next codon is CGU, and here it is, arginine. So far it makes sense in that what we're finding in the messenger RNA is exactly what we'd expect if we know the sequence of the protein ahead of time. And likewise, the third amino acid is lysine, and so we expect a codon for lysine to follow, the AAG. I bet we'll find that here somewhere. Here it is, lysine. So far it's exactly what we would expect. Now, the next amino acid is one called leucine. And so we'd expect the next codon to be a codon for leucine, GUA. But if we look, that doesn't appear to be correct because the fourth codon is GUA, which is a codon for amino acid called valine. But that's not part of the protein that we get out of this messenger RNA. So something seems wacky here. Why don't we get a valine amino acid if that's the next codon? And where is the codon for leucine located? Well, if we go looking around for the codon for leucine, it doesn't show up until over there. And so here's codon number 1, codon 2, codon 3, and then codon 4 is over there. This stretch of the pre-mRNA right here, these G's and A's and U's and C's, is never translated into any amino acids. You know, they look like codons, they're still G's and A's and U's and C's, but they're never going to be turned into any amino acids. And that's what introns are. They're segments of the messenger, of the pre-mRNA that, even though they're still part of the pre-mRNA, they never encode any amino acids. And just to finish off this protein, the next amino acid in the protein is GLU, so we'd expect in the messenger RNA a codon for that, glutamate, and GAG is it. And then the next amino acid is serine, so we'd expect a codon for serine to come next, but again, we don't find it right after that one. There's a gap of G's and A's and U's and C's before we get to that one's codon, and then there's the next codon for glycine, and then I guess a stop codon downstream because that's the last amino acid. So these segments of the pre-mRNA that aren't codons, they're not encoding any amino acids, are called introns. Segments of non-protein coding, in other words, non-translated nucleotides that are interspersed with the actual protein coding, translated regions of the messenger RNA. The segments, let's see if I have intron for that other one over there. There it is. So those are the introns on this messenger RNA. The segments that actually have codons that do encode amino acids are called exons. So there's the first exon, there's the second exon, there's the third exon. In describing this particular messenger RNA, we would say that it has 3 exons, 1, 2, 3. And again, those are the ones that actually have codons that the ribosome is going to turn into amino acids. And the segments that are still G's and A's and U's and C's, but are not turned into any amino acids by the ribosome, are the introns. So you'd say this mRNA has 2 introns and 3 exons. So these introns are very interesting things. If they leave you a little confused, you're not the only ones. When biologists first discovered them, they said, well, that doesn't make any sense, because the whole point of having messenger RNAs and even genes themselves is to encode proteins. Why have a bunch of nucleotides in the sequence that aren't really encoding any proteins? To make, I guess, kind of a strained analogy, if you read articles in magazines, sometimes you're reading an interesting article, but then they have a bunch of pages of advertising in the middle of the article, and you have to sort of just skip past those pages because it's not any information